Let's get started now. So today I'm really honored to present our ongoing study, this TenK10K multiome initiative, which is titled Genetic Regulation of Cell Type Specific Chromatin Accessibility Shapes Immune Function and Disease Risk. I would spend a few minutes at the beginning to introduce the background and the motivation, and a short introduction to our TenK10K cohort, and specifically to our TenK10K multiome project. So I'll start by introducing the concept of the complex diseases. So complex diseases are caused by a combination of genetic, environmental, and lifestyle factors. As you can imagine, there's all kinds of different factors play a role that contribute to the complex disease risk, like genetic effects, modifiable risk factor, like the BMI, physical activity, and also environmental effects as well. So particularly, we're interested in how can we identify the genetic association with the complex diseases. So about like two decades ago, so they come up with the genome-wide association approach, which is like a hypothesis of three methods that you collect a large scale patient samples and non-patient sample and collect their DNA and do the genotyping or whole genome sequencing. And you compare the frequency of the variants across whole genome between these cases and the controls, then identify the genetic variants that are associated with the disease phenotype. So after like 2 decades efforts, when people have identified hundreds of thousands of different genetic hits with a different complex diseases or trait, we found that most GWAS hits are located in a non-coding region. If you look at this pie chart, you can see that there's only around like 10% of GWAS hits located in the coding region, which means that the majority of the GWAS hits are just located in the non-coding region, making it really hard to identify or interpret their functional effects. So then it comes with this concept or technique called expression quantitative trait locus as intermediate effects in the SNP and trait association. Basically, you can collect the genetic information from the cohort and also at the same time measure the gene expression. So you can build a regression model between the three group of genotypes, then assess if the genetic variation or genetic mutation is associated with the gene expression level. Then you can overlap with these eQTL signals with your GWAS signal to see if it can help you to interpret the GWAS hits. However, there's multiple reports find that there's only 5 to 4% of trait associated overlap with the eQTL. And this has been described as a missing regulation puzzle. And people are really interested in like, how can we interpret this non-coding variants? There are different hypotheses why there's limited overlap between GWAS and eQTL. It could be that the eQTL effect is cell type specific, as we know that the gene expression not be observed across all the different tissue or cell types. It can also be cell state, so the genetic effects only show their effect on the gene expression in a certain cell state. And there's many other different hypotheses. So like around like 4 or 5 years ago, so our lab has come up with a 1K cohort, which is a pilot study of the TenK10K. So we collect 1,000 individuals each with single cell RNA sequencing from 1,000 cells and the genotype data. So we built a large scale eQTL atlas in around like 14 different cell types in PBMC. And if you look at the highlighted region, this kind of describes the number of eQTL signal that are specific to a certain cell types. So the conclusion from the 1K, a major point is that the eQTL effects are highly cell type specific. So if you do not have a single cell samples, you will very likely to miss a lot of eQTL effects. However, when we're still trying to overlap the GWAS of these cell types with the eQTL, there's still a lot of like missing. Now we are wondering like why is that the case? So there are also a lot of remaining challenges that most eQTL study only focus on common variants. When I say common variants, it means that the minor allele frequencies are larger than 5%, and we usually only test single nucleotide polymorphism and genome mutations. And also, because the limitation of the statistical model, usually when people map those single cell data, they have to aggregate the cells per cell type to make a pseudobulk, then using a bulk approach to run the QTL. And also, there's also other interesting intermediate molecular phenotype like chromatin, histone, and methylation are not evaluated for the same cohort. And also, what functional elements like promoter or enhancer are contributed to those genetic regulation are really underexplored. Last but not least, most of the association is built on specific genetic region. And in a genetic region, there are many SNPs are in correlation. We call the linkage disequilibrium. And it's really hard to link the association and to find the actual causal variants or the causal effects. And in the multiome study, we're trying to address the last 3 bits, but there are also other projects aim to address the first 2 points. Okay, so let's proceed with the TenK10K cohort. So this TenK10K cohort aim to map 5 million cells with around 10,000 individuals in Australia. So there are 5 main components of this cohort. We have 5,000 individuals from general population, 2,000 from heart disease, which is from the bioheart cohort, 500 for cancer, and 1,000 with autoimmune disease, and another 600 with COVID. So, so currently we have spent the last 2 or 3 years generating the phase 1 data, which are 1,000 individuals with the general population and 1,000 individuals from the heart disease population. This is the description of our phase 1 data. So we have around 2,000 individuals, and we collect their blood samples. And we also do the whole genome sequencing data for everyone. And for the PBMC data, we have 2 data sets. One is the RNA sequencing, and another is for ATAC-seq. Because we have the whole genome sequencing, we not only can run the association between the SNPs with the RNA and ATAC, but also other type of structure variation like short-time repeats, and as well as some somatic variants. So I will be talking about the multiome project today, which is mainly focused on identifying the association between the SNPs and the chromatin accessibility, and how to use this data to infer functional elements and identify causal mechanisms. So here lists several aims of this project. First, we're trying to understand the genetic effects of the genome regulation with a focus on intermediate phenotype, which is the chromatin accessibility. And also we're trying to identify the cis-regulatory elements and understand their role across different cell types. And one very important thing that we're trying to explain why there's a limited overlap between the GWAS and the QTL signals, and can we use chromatin data to address this limited overlap. And then by incorporating different layers of multiome data, can we identify causal variants for the disease association? And last but not least, when we have the multiome data, can we improve the gene regulatory network inference? I'll start by introducing the concept of chromatin. So chromatin is a genetic material that are kind of like folded in a 3D dimension that pack DNA, RNA, and the protein as a kind of formation of the DNA. So chromatin accessibility, same as RNA, is a quantitative measure. So the chromatin can be closed or partially open, and it also can be fully open. But imagine that if we have a single cell chromatin accessibility data, and if we look at the panel A, in different functional region like enhancers for a gene, then from the bulk data, you will have a peak signals, which kind of indicate that this chromatin region is open. But when you have the single cell data, you have the power to dissect this openness in different cell types. Like many times you will see, okay, the peak is only specific to a specific, open to specific single cells. So this helps us to understand the genome regulation in a cell type specific manner. So in the TenK10K multiome project, we collect around 1,000 individuals and using a ATAC-seq to process their chromatin accessibility data, and also multiome sequencing for small samples as a validation. For all these 1,000 individuals, we process their whole genome sequence data and measure, kind of call their SNPs and the indel variants. Indel stands for the insertion and deletion variants. And then with this, the other quality control, we run a series analysis to address the questions I mentioned above. So first, I'll talk about the data set a bit specifically. So for the main data set, we have around 922 donors after quality control. So in total, we have around 3.5 million nuclei. And in each cell types, we call the chromatin peaks. And in total, we have around 400,000 chromatin peaks. And then we're trying to test the genetic association with each peak. At the same time, because we also process the single cell RNA data, so we can map the same donors with their RNA. But I want to emphasize that this is unpaired multiome. So the RNA and the ATAC, although it's measured for the same donors, but they are from different cells. And then we also process around 60 paired multiome data from different cohorts, and we will use this ATAC-seq data as a replication cohort to validate the findings we identify from the major cohort. So, then the next important thing that we want to identify the chromatin accessibility QTL. So what is the caQTL? So if you collect a population cohort and have their genetic variants and the chromatin accessibility data, same as the eQTL, you can trace the chromatin accessibility at a phenotype and run the regression across the 3 different genotypes. And in many cases, you identify the significant association only in one cell type, but not in another cell type. And when you further look into the genetic different genotypes, you'll find that chromatin accessibility is different. And then it can help you to link the low accessibility and high accessibility to a different functional region like promoter, and to further understand the gene expression patterns. So in this data set, we conduct the caQTL mapping in 920 donors across 28 cell types. And we perform analysis using a TenK10K software with a GPU implementation. Because as I mentioned, there's a 400,000 peaks. And if you test all the SNPs within a certain region, it'll be really slow with the CPU, but with the GPU, you can finish one cell type within a few hours. To adjust the confounding factors, we adjust sex, age, genotype PCs, ATAC PCs, and library size in this association model. We also did some permutation to perform a permutation correction for false discovery rate. Then we identify a significant caQTL at the 5% FDR rate. So altogether, we have identified around more than 200,000 caQTLs across 26 immune cell types, corresponding to around 100,000 unique peaks. As you can see that the number of caQTL detected for different cell types varies a lot. But when we check out the number of caQTL with the nuclei per donor, so you can see a kind of very clear linear increasing patterns, which tells us that the power is kind of heavily dependent on the number of nuclei. And especially for the rare cell type, we're still lacking power to detect their caQTL signals. And we should keep increasing the sample size, especially for the rare cell types. So there are only 2 published studies around the caQTL, which is the first 2 panels. But the sample size is pretty limited. One is around 80 donors, one is 48 donors. And we try to replicate the caQTL signals compared with their signals. And you can see that the replication rate is very high, range from 75% to 90%. And we also have our in-house replication cohort. Although for the abundant cell types, you see a very high replication rate similar to the published study. But for some cell types, replication is low. And this is just because we have limited number of nuclei in these cell types. And for these cell types, there's only less than 10 nuclei per donors. But in the published study, usually they have around a few hundred. As I demonstrated before, so because the detection power is heavily dependent on the number of nuclei per donor. So I think this is just a power issue. But if you look at the most abundant cell types, the CD4 central memory T cells and the monocyte, the replication rate is really high. So after we get the around the caQTL signals, we really want to see if we can link those chromatin peaks to the genes and understand what's their functional role of their nearby genes. So we use a technique called colocalization. They help us to understand whether two phenotypes share the same causal variants. So if you look at the panel on the right-hand side, and imagine the red color and blue color, the eQTL and the caQTL signals. So there are multiple scenarios. So in the first scenarios, you see a strong signal in the eQTL, which means that genetic variants are associated with the gene expression, but there's no signals with the caQTL. At the same time, you will see some like different distinct peaks from the eQTL and caQTL. What is really interesting in this H4 scenario, which that the caQTL and eQTL signals overlap perfectly, and then usually indicate that they share the same causal variant. So by running this analysis, we identify 86 sound significance colloquial events involving with 31 sound peaks, and these peaks are considered as candidate cis-regulatory elements. So since this is only colocalization, so there's no like estimate for their effect size or direction. So we don't know if the peaks are upregulated or downregulated their nearby genes. But then we're looking for if there's any technical tools that can provide a causal support. And we utilize a framework called miniaturization. So short for MR. So this MR framework is used in the like epidemiology field. So usually it's used to study if exposure like smoking or other behavior has a causal effects for the outcome like lung cancer. And you can use a genetic variant as an instrument to estimate the effects of this exposure on the outcome. And this framework has been proven that it's free of the confounder that has both effects on the exposure and outcome. And imagine what if they are molecular traits. So in our case, we treat the exposure as chromatin accessibility and outcome as gene expression. So like in this scenario, we have the caQTL signals, which link the genetic variants to the exposure. And also we have the eQTL signals, which is association between genetic variants and gene expression. Then we can estimate if there's causal variants between this chromatin accessibility of certain peaks on the gene expression. So we test around like 800,000 peak gene cell type combination, and only limit to the peaks that only show the strong caQTL signals. And we identify 700,000 significant association. So trying to say, okay, now we are able to infer the cell type specific regulatory mechanism because we run this causal inference in different cell types. And what can we learn from these scenarios? So this figure indicates caQTL and eQTL colocalization and causal inference results. And we can start by looking at the first layers. So this is the peaks we're interested in, and the caQTL effects in the NK cell and dendritic cells. So if we look at the trend, so the minor allele has increasing effects on the NK cell and dendritic cells. But if we look at the bottom layers, which is a nearby gene, which is called MARCH8, the eQTL effect. And we found that in NK cells, the same allele provide a negative effect, but in the dendritic cells, it's increasing. So which means that if we link the peak to the genes, in the NK cells, the peak has a negative effect on the gene, but in the dendritic cells, it has a positive effect on the genes. And if we zoom in the genomic position, and the top two variants of these two caQTL are both located in the intron variants. And if we zoom in this gene, and we found that the chromatin peaks, which highlighted by the vertical gray line, is also located in the intronic region of the MARCH8 genes. And the MARCH8 genes has been reported to involve in some immune response. So we can further analyze the peak function on these genes with this causal effect support. So we are really confident about this causal inference. If you look at the one, and they all gave both a very significant value of 0.99, which indicate that this peak has a causal effect on these genes, and the effect has an opposite direction in different cell types. So since we have a robust inference from picked genes. So we next trying to answer the question, what is the colocalization with the GWAS results? Can we identify more overlap between the GWAS and the CQTL rather than using the eQTL? So as I mentioned in the first few slides, there's a limited overlap between the GWAS and the eQTL colocalization in the previous study. So we conducted colocalization between the GWAS and eQTL, and at the same time between the GWAS and the CQTL, and compare the proportion of the GWAS hit that colocalized with eQTL and CQTL separately. And we analyzed 16 different complex diseases and also 44 blood traits. And other than the colocalization, we also performed SMR, which is causal inference. And surprisingly, we identify across these 60 different traits, we identify around like 10% to 30% more CQTL colocalized with signal compared to the eQTL alone. If you look at this scatter plot, the y-axis is the percentage of the GWAS colocalization with the eQTL, and the y-axis is the colocalization with CQTL. For almost all the traits with the CQTL colocalization, you identify more GWAS hits. And the pattern is consistent between the colocalization results and the causal inference results. And then we try to understand why this, how to understand this missing regulation puzzle. So if we decide to look at this, we want to divide it, the colocalization pattern into 4 scenarios. So in this stacked bar plot, there are 4 colors, which indicate that if the QTL is colocalized with the GWAS, or there's only eQTL colocalization or CQTL colocalization. And if we look at the specific, this light blue region, which is the inflammatory bowel diseases, and you can see around like 10% of the GWAS signals only colocalized with the CQTL. And we want to understand why this case. First, because the QTL analysis is usually done in a fixed region, which means that for a certain gene or certain peak, you only test the genetic variants within, let's say, 100 kb or 200 kb or maybe 1 megabytes region. And if the functional elements like the enhancer is quite away from gene body, and this type of regulatory effects could not be captured. And if we look at this locus, and when we visualize the GWAS of the IBD signals, the y-axis is the minus log the p-values of the association of the SNPs. And you will observe a very strong signals in the interjecting region. It's not located in any gene. And if we overlap with the 100 kb eQTL of this gene, ETS2, and the colocalization results is nearly 0, which means that they are not overlapped. And in this case, in the pre-study, this is reported as not colocalized. But if you look at the peak, which is located in the interjecting region of the CQTL signals, you found a perfect overlap between the GWAS signals and the CQTL signals. And the vertical line indicates the location of the peaks. And this number indicates the probability of the colocalization, which is 0.9, is really high, close to 0.1. But if you're extending your eQTL search to 1 megabytes, and you will recover these signals. So this tells us that a lot of signals are kind of actually missed by a fixed cis region. But if we use the CQTL signals, even with a very narrow window, like say 10 kb, you can still identify this colocalization pattern, and then you will find this functional region. So this gene has been reported to involve in a macrophage inflammation. So we want to understand what's the function of these genes. So just as a study published last year, so they found that when they process with the ChIP-seq data, this is the monocyte specific. And if you look at the region, it's just located in the injected region. And they found that in the macrophage, there's a promoter and enhancer interaction. And the most significant peaks, if you look at back to our results, is in this region. And they did some like functional validation. So they used CRISPR to knock out this region and to see what's the effect of this gene expression and some downstream effects. So after the culturing of the primary monocyte to the macrophages, they found after knocking out the enhanced region, these ETS2 genes expression slightly become lower. And they also measure some like pro-inflammatory cytokine like the IL-1 beta, IL-1/6, and they all decrease after the gene expression of the ETS2 goes down. So this kind of validated that this gene is required for the macrophage inflammation, and the enhancer region of these function elements is the causal, is kind of causal for the genome regulation. Okay, so now we have proved that, okay, using the caQTL and the eQTL and GWAS can help us to link the chromatin peaks to genes and estimate the effect in specific cell types. And then what about the causal variants? So when we study a genetic risk of the genetic risk factors for disease, so we usually think about this way. So we have a genetic variants, effect the functional elements, which and consequently affect the gene expression, then increase or decrease the disease risk. So in our project, we have identified hundreds of thousands of caQTLs and also the eQTLs. And then we can also use the public GWAS. So when you combine the caQTL and eQTL, you identify those cis-regulatory elements. And when you combine with eQTL GWAS, you identify causal genes. But the question is that usually we only test the region, but there's maybe hundreds of thousands SNPs or genetic variants located in that region. And what is the causal variants? So there's a very latest techniques which can combine the GWAS and different multiple traits to identify causal variants. But I don't have time to go through the technical details. But basically, you can test multiple traits and to identify if there's a causal variant affect one or multiple traits and estimate the effect. So this is a visualization of this fan map results. So we test the inflammatory bowel disease as we demonstrate with the peak to gene links. We identify around 671 credible sets, which contains the causal variants. And among them, around 400 are single variant credible set, which means that we're pretty confident that there's only one variant that are causal variants. And I want to highlight this causal variants for the ETS2 samples, which is located in the interjected region we mentioned before. And if we zoom in this the SNPs and estimate the minor allele, which is the A allele, it has a decreasing effects of the chromatin peaks and also the gene expression of these ETS2 genes, and at the same time the IBD disease risk. And if we look at that study I mentioned, using functional experimental to verify the effect. So if you look at their results, so they did some ATAC-seq data in a monocyte, and this A allele, as I mentioned, that this is very consistent with our statistical inference. So the A allele is corresponding to a lowly accessible in this enhancer region. And they have done a really rigorous statistical test to verify this. And this effect is only specific to monocyte. So by looking at this example, I hope I convinced you that we're able to identify functional elements and the causal variants by incorporating the caQTL, eQTL, and GWAS signals. So now we have used all this data to infer the regulatory mechanism from the genetic variants to the gene, to the disease. But then we're also wondering, when we have the multiome data, can we infer the gene regulatory network, which help us to understand what's the gene-gene interaction or their effect. So the object of this part of analysis is to incorporate both paired and unpaired multiome data to infer gene regulatory networks. And also at the same time, can we integrate the QTL result to enhance the inference of gene regulatory network, identify potential upstream regulator. So we used a machine learning based framework called GLUE. So this framework can help you to integrate both paired and unpaired multiome data, but at the same time input some QTL information to guide this framework and help you to improve the network inference. I want to just showcase one example. So this is the peak to gene inference in the CD14 monocyte. and we use a specific eQTL to improve the peak-to-gene inference. And if you look at the graph, so each dot indicates a peak-to-gene path within the CD14 monocyte, and the orange color indicates that the inference score, which after we feed the eQTL information. And if you look at the orange part of this on the right-hand side, you can see that the inference score is significantly higher than those peaks without the eQTL inference. The pattern is very similar to if we use other cell types like CD4 naive cell types. And then since we have enhanced peak-to-gene inference, and if we put this peak-to-gene relationship into the gene record network, can we identify additional gene-gene interaction? The answer is yes. So when we incorporate the caQTL and eQTL information, and we identify more than 100 additional transcription factor and target gene paths, which is in 22 increase compared to the baseline inference without any QTL information. So this is an example that we identify from a specific networks, this APOBEC3A gene, and it is not identified when we didn't include the caQTL and eQTL signals. And this gene is as a target of upstream transcription factor IKZF1 and STAT1, and both transcription factors are druggable with specific approved drugs. And these genes has recently reported to be associated with breast cancer. And so these results show a really great potential to test some like repurposed drugs to fund this functional study of this gene. So I think that's all the results I'm presenting today. The conclusion that is that in this study, we present one of the largest single-cell atlas of chromatin accessibility in PBMCs, and we identify over 200,000 caQTL across 28 different cell types. And by integrating the caQTL and eQTL, we can identify candidates cis-regulatory elements and then link them to the target gene. At the same time, we can run MR framework to provide the causal support and estimate the effect size and the direction on their nearby genes. A very important finding from this gene is that we found that caQTL uniquely colocalized up to 30% of previously uncolocalized GWAS loci, and is revealed distal and cell type specific regulatory mechanism, which are missed by a previous eQTL study. So by incorporating all the layers with GWAS signals, we find map more than 1,000 causal variants, which affect chromatin accessibility, gene expression, and disease risk. And last but not least, a very important finding from this is that we can actually incorporate genetic information as a prior to improve the GI inference and reveal new TF target gene interaction. So as I introduced like in the beginning, there's a lot of remaining challenges. So in this study, multi-omics project, we only address these 3 questions, but we also have another 4 projects in the TenK10K phase 1 to answer the first 2 questions. So there are 5 studies led by 6 people. So Anna is focused on modeling a single cell eQTL and identify rare association between the gene, between the genetic variants and the expression. Hope is focused on the structure variation, which is a tandem repeats and how this type of variants affect gene expression. Albert at the end, they're interesting to identify causal genes and also what's their functional in the causal relevant cell types. Jenny is working on somatic mutation and study how clonal hematopoiesis affect the gene expression in different cell types. So we have a TenK10K publication bundle, and the first 2 papers are already on the bioarchive, and you're welcome to search them. Our study in the multiome and the causal genes will be posted online this week, and the title will be exactly like this. And if you're interested to see the full text of this multiome project, you're welcome to search maybe after this Friday. And there are also other more TenK10K projects coming. So I would like to thank a lot of people, especially Joseph, Daniel, Jama, and Alex for contributing and leading the TenK10K project and design the sample collections. And also specifically to Jaden, Fan, Oscar, Lawrence, Peter, and Blake, and working on the core part of this multi-omics study.